three.
Thank you. Thank you all. Hello, as I said, I'm Alida Palmisano, and I'm a research fellow at the National Cancer Institute. In my branch, we develop software to support biomedical research in the area of personalized medicine. So what I'm going to present you today is a tool that we recently developed called the D3Oncoprint, which is a standalone tool to visualize and dynamically explore annotated genomic mutation files. So to this audience in particular, I don't have to give you much background on the fact that we really need tools that are able to handle the increasing amount of genetic information and to be able to explore basically this data. But what I wanted to underline here, and what has been already mentioned several times in all the keynote actually today, is that what we found is kind of a gap between the people that develop the tool and the people that want to they have the data and they want to use the tool. So what we found in particular with our collaborator is that they had this specific visualization tool, which is called an Oncoprint. I will get to back to that in the next slide. And they wanted to use that, but that was not interactive enough for them, so they came back to us for questions to how to handle that. So just to give you a brief background, if you don't know exactly what an Oncoprint is, an Oncoprint is a very simple and effective visualization tool for mutations file. In particular, we are dealing with cancer research. So genetic mutation is like single nucleotide polymorphism, insertion, deletion, that kind of things. And a very useful way to visualize a big cohort of sample is basically a heat map. An Oncoprint is a heat map where each row is a gene, each column is a different sample or a different patient, and each cell is colored according to if that patient or that sample has a mutation in that gene. And different colors may mean different mutations like insertion, deletion, copy number variation, etc. But the point is, it's all in one single image, and in this way, humans and researchers basically that don't have like the bioinformatics capability skills, they can immediately see patterns if these, for example, there is a gene that is mutated in a bunch of samples, and these samples are a specific phenotype, for example, a specific stage of the disease, or like demographic kind of information like gender and things like that, then you can see a pattern in your data. The problem that our collaborator had with this specific tool is that, okay, we want to do this with our data. So how do they do that? The CBio portal offers a web oncoprinter, so basically you just upload your data there. But the web option has limited capability because, of course, it depends on what is the status of the implementation currently of the web interface. And in particular, with our specific data set, the problem is that you have to upload your data there. And there are two problems in that. First of all, the format is sometimes, in this case, for example, is not directly the output of a bioinformatics pipeline, like a VCF file. This is a specific format that they need to create the interface. And also, you have to upload your data, which is something that not all researchers want to do, especially at the beginning of their study. So, of course, this is something that the researcher that developed the Oncoprint are well aware of. And so they developed an R package, which basically you can run on your local machine. But that has the issue that it's an R package. So you need someone that has the bioinformatics skills to take the data and basically code this one to create, again, the Oncoprint that you want. So basically we stepped back and said, okay, these are very good things, but what is really an Oncoprint? So an Oncoprint is rectangle, text, and color. So that is basically the core of an Oncoprint. So what we decided to do is, how can we actually do the dynamic and interactive capability, easy customization, so with colors and movement and like order, and sorting and filtering, and have the initial data in a format that is the end of the bioinformatics pipeline of variant annotation. So this is what we developed. So the D3Oncoprint is basically a Java application that you can download on your local machine, so you don't have to upload anything anywhere. And it takes files that are in common annotated variant files, so either VCF file or tab delimited file or MAF file, which are the files that are used for TCGA data, for example. And the point is, with just two visually pretty simple, just click buttons and drop down kind of thing, you can then generate a Oncoprint, which is created in a web browser, so it's all dynamic there. So I don't have time to go to all the details of the specific format and specific options, but I have a poster at B751, so you can come there and we can talk more about the details later. But basically, this is a list of the main features, so it's a very simple interface, so anyone without any programming skills can use that. And it creates this HTML page with an embedded D3 Oncoprint and a data table right below that are all dynamic, and they can be filtered, sorted, and so the researcher can just interact directly with the data. We also added some hotspot to a specific database that have clinical importance, because our collaborators wanted to be able to immediately see if their sample have a mutation in those specific database. And all operations, again, are executed on your local machine, so there is no upload on any other server. So this is just the link to where to download it, and you just have to put your information, and then it's just a Java file, a jar file. So this is just, again, I'm going to go very quickly through the interface. So basically, in the first tab, the user opens it, gives the project a name, selects the folder where the output need to be, and just basically tells the program where the files are. They can be all in a single document or in a folder. And then there is an optional phenotype file, which basically characterizes the different samples, so that if you have information about, I don't know, demographic or stage of the disease, that can be used later for sorting. Then in the second tab, basically, the user will tell the software where the different information, the key information are, so the gene name, the variant, and what are these information. And these are all optional things that can appear in the data table later. So these are just optional information that are in the VCF file or in the annotated file and can be displayed later, but it's all optional. And so with the click of a button, this is created, and all this thing is in a HTML page, so all this is dynamic. So the color are, the things are colored according to if there is a mutation, each row is a gene, each column is a different sample, and all these that are the phenotypes are buttons. So the user can click and further basically interactive explore the data set. And on the top there is this Oncoprint, and with this button to change any basically visual aspect of this map. And right below, there is the same information in a tabular format, because in a text format it's much easier to do filtering and click on different information from external databases. So this is basically just what you can do with the software. You can just sort according to these different phenotype and find patterns in the data that are not easy to see otherwise. And here I just put like a screenshot of all the different capabilities that you can basically customize. Again, all one click away, pretty much. So this is my poster, and we can talk more about that over there.
Thank you very much. Yes, thanks very much. We have time for one question. Does anybody want to ask a specific question, or you? Yes, please, thank you.
So I noticed on one of the bullet points on the other data sources that were included, you had the user-defined bullet point.
Yep.
How easy is it to incorporate a user-defined format?
User-defined is the user-defined file for the hotspot variants. So it's basically a way to tell the D3Oncoprint to highlight a specific mutation that you want to see in your data. So for example, if this hotspot is of a specific cohort and you know that all the samples it's critical to see which samples have that specific variant, then you put it in a text file, and that basically is one of the input of the graphical interface at the end. So it's for the hotspot.
Okay, thanks very much.